So today I'd like to take a deep dive into a few experiments around ADCs that help researchers like you predict their stability once they leave the early discovery and development phases and move into stricter optimization, testing, and manufacturing phases. By doing this type of work early, you can de-risk the process of building ADCs by optimizing the conjugation process and formulation buffer. But first, I'd like to introduce a tool that will help you do all this. The Prometheus Panta is an easy-to-use instrument that gives you high-resolution data about your ADCs while using a minimal amount of sample. Among the benefits, the Panta offers precise measurements and gives you high-resolution data so you can detect subtle differences between your candidates. Label-free detection, which gives you flexibility in the types of buffers you can use and prevents any dye artifacts. Accommodates a broad range of sample concentrations, meaning you get predictive data about your ADCs without needing highly concentrated samples. And low sample consumption, so you can get multiple stability characteristics about your ADCs with just a small amount of sample. Now, the Prometheus Panta is able to achieve this by combining 4 different technologies that take measurements simultaneously across a single thermal ramp. The first of these technologies is nano-DSF, which uses your sample's intrinsic fluorescence to assess thermal stability. Next, we have DLS, which is used to check the purity of your sample and measure aggregation. Third is back reflection, which provides turbidity information that can help to tell you if you have large aggregates forming in your sample. And last, we have SLS, which can resolve small changes in size and detect small aggregates. Now, I won't be going into the details of how these work together in this talk, but if you're interested, you can learn more from the Explore the Prometheus Panta video. There are a lot of parameters that are directly measurable by the Panta, and several more that can be derived from the data. The ones I'll be focusing on today are highlighted here, and these will provide predictive stability characteristics that were measured by analyzing ADCs with the Prometheus Panta to assess self-association, kinetic stability, and isothermal stability. Now, before I get into that, I'll first give a brief overview of ADCs. So ADCs, or antibody-drug conjugates, are exactly what they sound like: a monoclonal antibody connected to a small molecule drug by a linker. Conjugation of the small molecule to the antibody and subsequent production steps can affect antibody stability in a number of ways. First, the proximity of the small molecule to the antibody will change the antibody's chemical environment, affecting its stability. Second, the conjugation process itself may require buffer conditions that are destabilizing, especially if the reaction requires a long incubation time. Cleanup processes are vital to filter out any remaining unconjugated molecules in the solution that could destabilize the ADC. And the final formulation must be optimized to increase stability of the ADC. It's important to remember that the best buffer conditions for the naked monoclonal antibody may not be ideal after conjugation. So comparing the ADC to the naked antibody is a very helpful way to determine whether the conjugation process has had too much of a negative impact on its stability. And the Prometheus Panta gives you high-resolution stability data to help you do just that, and lets you identify stability impacts down to the individual domains. So today we'll be comparing data from 3 biologics: Herceptin, which is a monoclonal antibody; Kadcyla, which is an ADC derived from Herceptin; and disitamab vedotin, which is also referred to as RC48. And I'll be using this name as it's considerably easier to say. This is not quite a one-to-one comparison. While these therapeutics all target HER2, RC48's naked antibody is hertuzumab, not Herceptin or trastuzumab. Compared to Herceptin, disitamab vedotin has a higher affinity for HER2 and shows more antibody-dependent cell-mediated cytotoxicity in vivo studies. Another thing to note is that the conjugation of RC48 is via cysteine binding rather than lysine binding, which is the case in Kadcyla and all other currently approved ADCs. I'm going to be including RC48 here because it shows some interesting behavior that we'll get to in a little bit. Now, all 3 of these therapeutics have been optimized for use in the clinic, so the stability data is going to look better than if we were, say, comparing candidates early in the development process. Still, the results we'll look at in a bit are a good example of how the conjugation process affects the stability characteristics of your ADCs. So one way to compare the head-to-head stability of your ADCs is by looking at thermal stability, and nano-DSF provides an easy way to do so. By using the intrinsic fluorescence of a molecule, nano-DSF provides a label-free method to measure how a biologic unfolds across a thermal denaturation gradient. And in doing so, we can identify melting temperatures, or TMs, associated with different domains of your ADC and compare these across candidates. The higher the TM, the more thermal stability a candidate has. This also gives us a way to quantify how the conjugation process affects thermal stability, which we'll look at now. So here we have a comparison of the conformational stability of Herceptin and Kadcyla, which is the ADC derivative of Herceptin. As I mentioned before, these are both currently available in the clinic, so they've already been optimized in terms of stability. But by using the Prometheus Panta, we can still detect some subtle differences between these two biologics. So the nano-DSF results in the top part of this figure show how the intrinsic fluorescence changes across the thermal denaturation gradient. And the bottom part of this figure is the first derivative view of that data, which makes it easier to visualize some of those key transitions. So these results, which come from an set of experiments run in triplicate, demonstrate how the Panta's reproducible data reveals important insights about small differences in conformational stability and how those changes affect individual domains. So these are all individual data points along an unfolding curve. Now these light blue lines show where the melting points of Herceptin, which is in the black line, are. We can see the peaks of the dark blue line, which is Kadcyla, are shifted slightly to the left compared to those of the naked monoclonal antibody, indicating that the ADC is less stable comparatively. Now, typically in a thermal denaturation gradient, the FC1 domain unfolds first, followed by the Fab region and then the FC2 region. The Fab and FC2 regions generally have similar melting temperatures that can't be resolved, so the second TM represents both of these domains unfolding. So we can see with the data here that the TM1 peak is shifted more than the TM2 peak, so we hypothesize the conjugation had the most impact on the FC1 domain. So now this is the same data, but with RC48 added in, and that's that purple line that you see. So we can see that the TMs for RC48 are even lower, indicating that RC48 has lower thermal stability compared to both Herceptin and Kadcyla. We can also see that the shape of the unfolding curve is different, which does make sense because the naked antibody for RC48 is not Herceptin. So this is just a quick example of how the data resolution provided by the Panta can help you to get a better understanding of how conjugation affects antibody stability. But now let's talk about more in-depth experiments that offer you predictive information about your ADCs and their stability. So we just saw the nano-DSF data, but as I mentioned earlier, the Prometheus Panta also uses light scattering to assess biophysical characteristics of biologics. One such characteristic is self-association, which describes the propensity of a biologic to become viscous or aggregate. These measurements are important to get a sense of how your ADC will behave at higher clinically relevant concentrations. So in early stages, we know you may be working with low concentration or low volume sample preps. So self-association studies are really useful for predicting how your ADCs will act at higher concentrations. This helps you to optimize your formulations for better stability at high concentrations, and also helps you to identify optimal candidates or buffers earlier in development and de-risk downstream cleanup. I won't go into too much detail about this technique since we do have a fair amount of data to discuss, but essentially we measure the light scattering of your samples at a few concentrations, plot those measurements, and fit a line to the data. Depending on the type of experiment, the slope of that line provides the KD or B22, which tells you about your ADC's tendency to self-associate. Now, a positive slope means your sample is less likely to self-associate. A negative slope indicates the opposite. So on the right, you can see we have a plot of data where we use DLS to measure the KD of NISTMAB in two different buffers, one with sodium chloride added and one without. So plotted in the black, you can see that the NISTMAB in the buffer without salt has a positive slope and therefore a positive KD. This positive slope is what we want to see, as the positive KD indicates that the antibody is repelling itself and won't aggregate or become viscous at higher concentrations. And we can see in the buffer with salt, the KD is negative, indicating that the NISTMAB is attracting itself, which can lead to problems at higher concentrations. Now, this is caused by how the salt is affecting the electrostatic interaction between the protein molecules. Just as a side note, this is a really good example that shows how important this sort of analysis is, because salt concentration, buffer pH and additives, and characteristics of the biologics themselves all interact with each other to contribute to propensity to self-associate, making this really difficult to predict without doing this type of analysis. So here we're looking at the KD of Herceptin. As you can see, the slope, and therefore the KD, is positive, indicating that the Herceptin molecules are repelling each other, so Herceptin does not have a propensity for self-aggregation. When we compare the KD obtained from our Prometheus Panta data to literature values, we can see that they align. But now let's compare the KDs of all three biologics we've been discussing. So we just saw that Herceptin has a very positive KD, which is great and means that the antibody molecules are repelling each other. Kadcyla has a less positive KD, but it's still positive, which also indicates that there is repulsion between the ADC molecules. RC48, however, has a negative KD, which means that the RC48 molecules are associating with themselves and may aggregate with the current buffer conditions. So based on these KDs, we can start to predict which of our biologics are more likely to self-associate. Now we'll look at the osmotic second virial coefficient B22. The B22 is measured with SLS as opposed to DLS with KD and describes the strength of protein-protein interactions more globally. Like KD, the slope of the line will indicate if the net protein-protein interactions are repulsive or attractive, with a positive B22 being predictive of a low tendency to aggregate and a negative B22 predicting the opposite. Like we saw with the KD measurements, Herceptin looks great, that's positive. With Kadcyla, the conjugation of the small molecule reduced the self-repulsion, but we still have a positive B22. RC48 has a negative B22, which again indicates that there's some self-attraction happening. And we're seeing the same overall trend that we saw with the KD measurements. So to summarize the results we've just seen, we can use the Prometheus Panta to predict how likely an ADC is to self-associate at higher concentrations. By looking at the slope of a line fitted to scattering data of different concentrations of your samples, we can obtain a KD and B22, which tells us about the propensity of an ADC to aggregate or become viscous at higher concentrations. Doing these types of experiments gives you important information about how your ADC will act at clinically relevant concentrations, and they don't require high concentrations or volumes that you may not have at this stage of development. It's important to note that based on the way Panta takes measurements, we're doing both the DLS and SLS measurements simultaneously, which means you can get a KD and B22 from the same experimental run. These self-association experiments require a little bit more setup, but they give you important predictive information about your ADCs before you scale up. Now, based on the way these values are calculated, the concentration of your biologic and the strength of other interactions occurring in solution, such as hydrodynamic interactions, these can result in KD or B22 being a more appropriate measure of propensity for self-aggregation. This is just something to be aware of as you start to plan these experiments, but fortunately both are easy to obtain in the same experiment. So now I'd like to talk about another type of predictive information that the Panta can provide about your ADCs, which is kinetic stability. And kinetic stability refers to how long a protein can remain active under a set of given conditions before undergoing irreversible denaturation. Now, we know that the long-term stability of ADCs is important when evaluating developability, but the timeline required to do these types of studies is incompatible with development workflows when you're still deciding which ADC candidates and formulations are best. But by performing accelerated stability studies that measure kinetic stability, you can get predictive information about long-term stability in just a fraction of the time, helping you to identify candidates that are more likely to have a viable shelf life. In the next few slides, I'll show you how we can use data obtained from the Prometheus Panta to calculate the half-life of 3 biologics we've discussed so far. So the model we'll be using takes into account how the heating rate affects melting temperatures. So the figures I'll share show a series of thermal denaturation ramps that use heating rates ranging from 0.2 to 6.4 degrees Celsius per minute. So this is Herceptin, and the red line here is showing the slowest heating rate, which is 0.2 degrees, and the purple is the fastest, 6.4 degrees. And we have a range of rates in between. And underneath, like the nano-DSF data we saw earlier, that first derivative view shows the TMs and how they're changing as the heat rate change. Now we can do the same thing with Kadcyla and RC48. Same thing, this is just showing a range of heating rates. So we'll use this data to calculate the energy of activation. So this uses that nano-DSF data we just saw, and we've generated an Arrhenius plot. And the slope of this plot is used to calculate the activation energy of irreversible unfolding, another predictive measure of long-term stability. Like the thermal unfolding data, we see that the conjugation has slightly reduced the kinetic stability of Kadcyla relative to Herceptin, but not by much. By comparison, the activation energy and therefore kinetic stability of RC48 is much lower, indicating that it's less stable, and this is consistent with the other stability data we've looked at. This is interesting because we're really seeing how the type of conjugation can affect an ADC stability. So remember, RC48 uses cysteine binding, whereas Kadcyla uses lysine binding. So from these results, we can conclude that the conjugation via lysines is actually better tolerated than conjugation via cysteines. So now we've taken the TMs and the activation energy of the 3 biologics we've been looking at and plotted them on a heat map to look at how these different parameters contribute to overall stability. So the x-axis is TM1, of the ADCs, and the y-axis is the energy of activation for irreversible unfolding that we just calculated previously from the Arrhenius plot. And when we plot our ADCs, you can see RC48 is down near the bottom left, and Kadcyla and Herceptin are near the top right, because both have more thermal and kinetic stability compared to RC48. So we can actually take the TMs and energy of activation and use them to calculate the expected half-life of the biologics. When we do, it reveals that RC48 has a drastically reduced half-life of just under 20 hours compared to Kadcyla, which is at 62 days, and Herceptin at 281 days. So by using the Prometheus Panta and varying the heating rate of thermal denaturation gradients, we've been able to calculate the expected half-life of our ADCs, helping to predict which ADC candidates will be more or less stable in the long run. So we've just walked through how we can take data from the Prometheus Panta and use that to get information about the kinetic stability of your ADCs. Now, this type of accelerated stability study takes a few more steps, but doing these experiments can give you the energy of activation for irreversible unfolding and the predicted half-life of your candidates. And by doing these types of studies, you can get a sense of the long-term stability of your ADC candidates without needing to perform long-term experiments. Now let's talk about the last predictive measure of stability, and that's isothermal stability. As I mentioned when we were discussing kinetic stability, assessing long-term stability of your ADCs can be a challenge. At this point in development, you need information about which candidates are most likely to be stable without spending a lot of time getting that information. Now, by holding ADCs at an elevated temperature just below their melting temperature for an extended amount of time and analyzing that scattering data, we can get an accelerated look at their long-term behavior. So here we have DLS data from our 3 biologics, Herceptin, Kadcyla, and RC48, held at 65 degrees Celsius for over 13 hours. Now, to orient you to the graph, we have time on the x-axis and the cumulant radius is on the y-axis. By using DLS to monitor the change in cumulant radius of the molecules over time, we can see when they begin to unfold and aggregate at this elevated temperature. So just as we've seen before, Herceptin, which is represented by the black line, is very stable, and we see a minimal change in the cumulant radius. Kadcyla, shown by the blue line, is less stable, which we've seen before, and this is attributable to the conjugation of the small molecule. RC48, however, shows a faster increase in the cumulant radius, which indicates that aggregation is occurring and indicates the lowest degree of isothermal stability. I'd just like to note that these results reinforce what we saw with the previous kinetic stability data. So we're consistently seeing that RC48 is less stable across all of the various stability measures we've looked at today. And this was a relatively simple experiment, but it still gives you valuable information for predicting how your ADCs will behave without needing to do long-term stability studies. So by holding your ADCs at a constant temperature for a few hours or even days, you can see which are aggregating and are more likely to be unstable in long-term storage conditions. And these types of studies are also really helpful to identify which buffer formulations will provide the best conditions for long-term storage. So we went through a lot of data, so I'd just like to do a quick recap about what we discussed and how it can help you screen your ADCs. The Prometheus Panta can give you multiple predictive measures of stability to help you screen your ADCs and predict how they behave, either in higher concentrations or in long-term storage. Now, at higher clinically relevant concentrations, we know there's a risk of ADCs becoming viscous or aggregating. Self-association experiments to calculate KD or B22 can give you insight into whether your candidates are likely to cause issues when scaled up for manufacturing and delivery. And the nice thing about performing these experiments with the Panta is they can be done with low amounts of sample, meaning you can screen your antibodies for risk of self-association before you scale up production. The Panta can also be used to do accelerated stability studies. These thermal experiments can save you valuable time by predicting which candidates are more or less likely to be stable in long-term storage conditions. Kinetic and isothermal stability measurements both give you valuable predictive information about the long-term stability of your ADCs and, as we saw, how conjugation impacts the stability.